Hello everyone, I'm Professor Lee of Dr. Anti-Aging and Herbal Medicine TV. Last time, we studied DNA damage, one of the fundamental causes of aging, and how to prevent damage. In part 2 today, we'll study the problem of decreased ability to repair damaged DNA and the accumulation of senescent cells. Let's get started. All of our cells have the ability to repair damage. The process of healing skin wound is a visible repair process, but the repair system always operates even at the level of small molecules that cannot be seen with the eye. About 100,000 DNA damages occur in human cells every day. Most are repaired, but as few as 10 and as many as 500 DNA damages remain unrepaired and continue to accumulate. Compared to a person who accumulates 10 unrepaired damage per day, a person who accumulates 500 damages will have a much different aging speed. This difference in repairability is not just due to aging differences, so aging has begun to be viewed as a disease. If damaged DNA is managed well for repair, age can be delayed. How to enhance DNA repair will be introduced in the next lectures. The amazing ability of our cells to repair DNA damage is called the DNA damage response. In 2015, the Nobel Prize in Chemistry was awarded to 3 scientists from Sweden and United States for their discovery of the mechanism for repairing damage that occurred during DNA replication and repairing DNA bases caused by ultraviolet rays or chemicals. This achievement has become an important basic research for treating cancer and neurodegenerative disease. In addition, strategies to enhance DNA repairability has become an opportunity to play an important role in extending lifespan and healthy aging, so a clinician like me can introduce you to a method for treating aging by enhancing DNA repairability. I'll briefly explain the process of repairing damaged DNA. The first step is to quickly recognize the damage, right? This function is performed by the damage detection proteins. They are so sophisticated that they are separate proteins that detect when the entire double strand of DNA is damaged or when only a single strand is damaged. The second step is to sound a warning alarm and call another repair team, which is performed by another protein. If the damage is too difficult to repair, it's not repaired and instead causes the cell to die. The final step is where real repair begins. Even here, different repair mechanisms come into play depending on the types of DNA damage. They cut out and repair damaged bases, correct errors in DNA replication, quickly and temporarily reattach broken DNA strand, and, when there is time, slowly and precisely reattach the cuts. Isn't that amazing? If any one of these has a problem, DNA repairability decreases and cancer incidence increases. In animal experiment, if only one device breaks down, repairability decreases by 70% and cancer incidence increases by 90%. When I introduce food, herbs, and pills that are effective in enhancing DNA damage detection ability, enhancing damage risk a lot, and enhancing recovery in the next third lecture, you will understand what I mean. From now on, I'll talk about what happens when damaged DNA cannot be repaired. First, the genome becomes unstable. If unrepaired damage continues among the 3 billion pairs of DNA, it's natural that the genes contained in it will also be damaged, and this is called genome instability. Genome instability is like the pillars of a building becomes weak. At first, damage occurs here and there throughout the building, and if damage occurs to the pillars over time, the unstable structure becomes more likely to collapse. If this instability worsens, various problems such as cancer and chronic disease can occur. In a study of 500 breast cancer patients, those whose tumor tissue tests show high genomic instability had an average of 35% faster cancer progression and a 20% lower treatment response rate than patients with the same breast cancer who did not have this finding. The second topic is today's topic, accelerated aging. Accumulation of unrepaired DNA damage is the main cause of senescent cells. This is because all cells with damaged DNA turn into senescent cells. What happens if we leave cells with unrepaired damage as they are? Eventually, they all turn into cancer cells. Therefore, our cells activate an amazing survival defense mechanism to prevent cancer cell development. If there are no such survival mechanisms, cells with damaged DNA would turn into cancer cells and spread throughout the body, and everyone would die from cancer. In order to prevent damaged cells from turning into cancer cells, the first thing they do is wake up the tumor suppressor gene. This protein is famous tumor suppressor protein p53 and is also nicknamed the guardian of genes. In fact, it's a characteristic of various cancers that the cancer suppressor protein p53 is not activated when it is needed. When the number of awakened p53 increases, it enters the nucleus where the damaged DNA is located, as shown in the picture, and wraps around the damaged DNA. Then the cells choose one of three survival strategies. The cell's desperate effort to survive are truly amazing. The first survival mechanism is that a specific gene activates to stop the cells for a short time. A cell that stops for a short time is not yet a senescent cell. During the short pulse, it gains the time needed for DNA repair and prevent damaged DNA from being passed on to descendant cells. The Nobel Prize in Physiology of Medicine in 2001 was awarded for discovering how the cell growth cycle is regulated by stopping and speeding up. The second option is that when serious DNA damage or irreversible genetic abnormalities occur, p53 causes the cell to die completely, preventing mutation from spreading in advance. It's better to die than to become a cancer cell. The Nobel Prize in Physiology of Medicine in 2002 was awarded to three scientists for discovering that this process of removing damaged cells is an essential process for life that's regulated by genes. The cells that die in this way are recycled by the autophagy system, which will be explained in another topics on aging treatment. The last third survival mechanism is when there is continuous DNA damage to the extent that the repair process becomes overloaded. That is, the cells stop growing and changes in the stationary state because of accumulated DNA damage. Rather than turning into cancer cells, it chooses to stop growing and become stationary but not dead. Cells that change in this way are called senescent cells, and the increasing number of senescent cells is one of the key phenomena of aging. Of the three survival mechanisms of cells with accumulated DNA damage, two have been awarded the Nobel Prize, and the mechanism of becoming a senescent cell has not yet been awarded the Nobel Prize, but is considered a strong candidate. Until now, aging was thought to be an inevitable process that everyone goes through as they get older. However, since aging is the result of survival defense mechanism that select to prevent damaged DNA from turning into cancer cells, the latest viewpoint in modern aging biology is that if we prevent DNA damage as much as possible and ensure that the repair system operates well, we can at least slow down the aging process. Now, let's summarize the key points of aging treatment caused by DNA damage. 1. Minimizing DNA damage, and I talk about the important lifestyle prevention method in the first lecture. 2. Strengthening recovery ability, which is the main content of the next third lecture. Third method is to remove the remaining senescent cells. Since removing senescent cells will be a new anti-aging treatment in the near future, this part will be dealt with separately in the Removing Senescent Cells chapter. Thank you for your hard work starting today. I'll see you again in the next lecture, part 3.